Hello, my name is Dr. Roderick L. Roll, and today I will be talking to you about staining bacteria. Staining a bacteria or any cell is going to increase contrast and resolution. This is going to be achieved by differences in structures which can be observed by coloring. The first thing you would have to do is create a smear. A smear is a thin film of microorganisms on a slide. The next thing you will need to do is air dry the emulsion on the slide. After the emulsion is dried, then you would need to either heat fix the emulsion onto the slide or chemically fix the emulsion onto the slide. Microbiological stains are usually salts, and they are composed of either a positively charged ion or a negatively charged ion. If it is positive, it is a cationic salt stain, and if it is a negative charge, it is a anion stain. Acidic, which are negative dyes, will stain alkaline structures. These alkaline structures will be positive. Opposites attract. The dye is going to give up a hydrogen ion, leaving the stain negative. Once the stain is negative, then it will stain alkaline structures. One example of this acidic dye is eosin Y. The basic dye is positive, and it will stain acidic structures. The basic dyes are the most common because the cell wall is composed of many negative charges. An example of a basic dye is methylene blue. This is the chemical that you use to stain the cheek cells in your biology class. The first thing you would need to do after you take this powder and mix it with water is to filter it. So you will filter this stain through a funnel. You want to make sure you filter it so that you can trap all the undissolved crystals into the filter paper. If not, you might accidentally see a crystal and think it is a microorganism. Chromogen is the color molecule of the dye. It consists of the chromophore, which is the color portion, and the oxochrome, which is the charged portion. So here is an image of the chromogen. Notice that you have two portions. The chromophore is the color, and the oxochrome is the charge. These dyes, these stains, are always going to be dissolved in a solvent. Sometimes the solvent is water, sometimes the solvent might be acetone or alcohol. So the dye is named for what it stains. So the acidic dye is negative, and the basic dye is positive. The basic dyes include malachite green, crystal violet, methylene blue, and saffronin. They will all stain acidic structures. These dyes are high in hydrogen ion concentration. Therefore, it will stain negatively charged molecules. This is the typical structure of a bacterial cell wall. Notice that the gram-positive cell has a larger, thicker layer of peptidoglycan. whereas the gram-negative has a very thin peptidoglycan. The acidic dye, which is negative, they include eosin and Congo red. These stains are going to stain alkaline structures. These dyes are high in hydroxide ions, so therefore it stains positively charged molecules. Several staining categories exist. We're going to talk about 3 of them. The first category is simple stain. The simple stain is composed of just using one basic dye. It is used to determine the shape and the size of the cell. The different shapes of cells that you will encounter is bacillus, coccus, and spiral. These are the common shapes of bacteria. So the spiral or spirilla or spirillum for singular is a spiral-shaped bacteria. Bacilli or bacillus for singular is a rod-shaped bacteria. Cocci or coccus, which is singular, is a round-shaped bacteria. A simple stain can also be used to determine the arrangement of cells. So the shape is bacillus, and it can be just one bacillus, or it can be 2, or it can be V-shaped, or it can all be lined up in to resemble a fence called palisades. Or it can be a long line of hundreds that will be called streptobacillus. So this is a depiction of how those might look. In image B, we see 2 bacilli together. This would be called diplobacilli. We see a long chain in letter C called streptobacilli. We see the palisades and we see the V-shaped. So simple stain can be used to see the size of the cell, the shape of the cell, and the arrangement of coccus can also be differentiated into many different arrangements also. Diplococcus, tetrad, sarcina, staphylo, and strepto. This slide shows you what it looks like. 2 coccus together is a diplococci. A long chain of circles is streptococcus, a streptococci. A 4 together is a tetrad, 8 sarcina, and a big cluster is staphylococci. Variations of the main 3 include vibrio, which is slightly curved, coccobacilli, which is a short rod, and spirochetes. These are flexible spirals. These are all the different shapes you might see after you simple stain the microorganism. The second type of stain we will talk about is differential stain. Differential stain contains more than one stain. There's 3 examples: the gram stain, the acid-fast stain, and the endospore stain. With the gram stain, both cells types will appear clear or transparent. You will then flood the slide with crystal violet. This is going to make gram positive and gram negative appear purple. Then you're going to use a mordant called iodine. This is going to help anchor the purple dye to the cell wall. The next step, you're going to decolorize, and this is going to remove the purple color from the gram negative bacteria. Finally, you will use a counter stain called safranin. Now the gram positive will appear purple and the gram negative will appear red. So this is step by step how to do it. Step 1. Slide is flooded with crystal violet for 1 minute. Then you will rinse with water. Remember, there's a difference in the cell wall of bacteria. The gram positive has a very, very thick peptidoglycan, about 40 to 50 layers. Layers of NAG and NAM. This is a modified sugar. Whereas gram negative has a very thin NAG and NAM layer, maybe only 4 or 5. So the difference in the cell wall is going to be exploited when using a differential stain. Step 2. The slide is flooded with iodine for 1 minute. This mordant is going to help the stain stick. Step 3. Slide is flooded with solution of alcohol mixed with acetone for about 10 to 30 seconds. Then you're going to rinse it with water. The resulting smear is decolorized. The gram positive is going to remain purple and the negative is going to be colorless. Gram negative bacteria have a high lipid content because of the outer membrane and a thinner NAG-NAM. So this is the major difference between gram positive and negative. The gram negative has a very thin NAG-NAM layer, and outside of it you're going to have another membrane. The alcohol acetone mixture is going to decolorize the gram negative by extracting the lipids in the outer membrane. So this is going to remove the color from the gram negative. Step 4, the slide is going to be flooded with safranin, which is a counter stain. Once this is done, you will then rinse with water and blot it dry. The gram positive will remain purple, whereas the gram negative will be colorless. But the safranin is the counter stain, which is going to then restain the cell red. The next differential stain we will talk about is acid-fast. It is also called Ziehl-Neelsen stain. So first, both cells are colorless. You're going to stain with a stain called carbofusion. This is going to stain all cells red. Then you're going to decolorize with acid alcohol, and it is going to remove the stain from the acid-fast negative cells. The acid-fast cells are going to stain red, and now you have to come back and counter stain a different color. So in this image they used brilliant green, which stained the acid-fast negative cells green. The acid-fast stain is used to penetrate the waxy lipid cell wall of bacteria like Mycobacterium, and Nocardia. Notice in the image you have a thick, waxy mycolic layer. This mycolic layer is composed of mycolic acid, a waxy lipid. Bacteria like Mycobacterium tuberculosis has a very slow replication rate. The doubling time can be anywhere from 12 to 20 hours. So it takes several weeks to visualize a colony on a petri dish. But that slow-growing bacteria can be stained with a carbofusion stain. The utilization of steam will have to be used to help drive this lipid-soluble phenolic dye into the mycolic layer. Acid alcohol is then used to decolorize the residual dye. This is followed up by a counterstain of methylene blue. In this acid-fast stain, you see that the microbacterium is going to stain red from the carbofusion, and the Streptococcus pyogenes is going to stain purple or blue. In this slide, you see the counter stain. It stained the Staphylococcus, whereas the carbofusion stained the letter B. The next differential stain is the endospore stain. This is the Schaeffer-Fulton stain. This is going to stain endospore producers like Bacillus thuringiensis or Clostridium. So these cells and spores prior to staining will be clear or transparent. After staining with malachite green, the cell is going to be green and the spore is going to be green. You would then decolorize with water, and then the cell will be clear and the endospore will stay green. Then you will counterstain the rod-shaped bacteria, Bacillus or Clostridium, with safranin. The endospore stain is used to visualize Bacillus and Clostridium. Examples will be the causative agent of anthrax, gangrene, and tetanus. The first thing you would do is you would need heat, and this heat is going to drive malachite green into the impermeable endospore. So the endospore will appear green, and the vegetative cell is going to be red. These stains can help differentiate organisms based on the location of their endospore. If it is on the end, the very end, it is called the terminal endospore. If it is almost at the end, it is a subterminal endospore. And if it is in the center, it is called central endospore. The third category of staining is special staining. We're going to talk about a negative stain, a capsule stain, and the flagellar stain. The glycocalyx is a coat of molecules such as sugars and proteins that are external to the cell wall of bacteria. So this is outside of the nag and nam. This is outside of the peptidoglycan. The glycocalyx can be either highly organized or loosely organized. If it is highly organized, it is referred to as a capsule. It is also tightly attached to the peptidoglycan. If it is loosely organized, and loosely attached, it is referred to as a slime layer. So the glycocalyx can be seen on many different types of bacteria, but depending on if it's loosely attached or tightly attached, it can be either a slime layer or a capsule. The capsule helps the bacteria avoid desiccation. It also protects the bacteria from ingestion and destruction by the white blood cells, like the macrophage. It also gives a defined border. So in the Griffith experiment, you remember that they had a virulent streptococcus and a non-virulent streptococcus. What made the one streptococcus kill the mouse was the presence of this capsule. When Griffith injected the mouse with the virulent strain, the white blood cells were not able to attack streptococcus because of the capsule. Whereas some of the mice were able to live because that bacteria did not have a capsule. So the white blood cells were able to immediately destroy that species of streptococcus. So sometimes you'll have the capsule present and sometimes you will not. The presence of a capsule in Streptococcus pneumoniae is the most important factor in its ability to cause pneumonia. Strains that do not contain the capsule can be destroyed by a macrophage. Bacteria with loosely attached glycocalyx is called the slime layer. This can be washed off very easily. So the capsule can be stained by the capsule stain or a negative stain. We will discuss both. The capsule stain will use a negative acidic dye to stain the background around the capsule, therefore leaving the cell colorless. Remember, the most common stains we use are the basic stains. The basic stains are used because it stains the cell wall. The cell wall is negative. So if the capsule is negative and we use a negative dye, it's going to stain the background around the cell wall, around the capsule. Most bacterial cells are negatively charged. Cell shrinkage and distortion is reduced because no heat is used in this stain. This is how to use the stain. Acidic dyes such as Congo red and nigrosin are going to be used to stain the background. Then a basic dye like Manavel is going to be used to stain the cell. The next stain is the negative stain. It also can stain the capsule. The only difference in this stain and the capsule stain is the interior cell is not going to be stained. In this image, you see how the negative dye is staining the background because the cell wall and the capsule is negative. We will now talk about the flagella stain. The flagella stain is going to stain a flagella. A flagella is an appendage to the bacteria that is going to help the bacteria move. It is a filament that is composed of cytoskeletal proteins. The location of the flagella is important. It can be on one end, it can be all over, or on both ends. If it is on both ends, it is called amphitrichus. This is an image of a flagella stain. Spirochetes, such as Treponema pallidum, is amphitrichous. It has one flagella that goes into the media and one flagella that stays closely attached to it. It is referred to as the endoflagella. Both of these organisms have flagella. They are spirochetes. Treponema pallidum is the causative agent of syphilis, and Borrelia is the causative agent of Lyme disease. They both have a endoflagellum, which helps them to bore themselves into the tissue.